starboard port too?
No, you can leave those off.
Full deck, that's Hercules past the transom.
Roger, turn it and burn it.
Copy, past transom.
Control deck, that is tether, all out.
Copy, tether all out.
So, as I predicted, I'm at the end of my tether and struggling to get over there. But close enough.
Okay, control deck, we have a Hercules center up in between the goalposts. You guys set for release.
Good, good. We're all good.
Control is ready.
I'm just going to bring Herc below the waves a little bit, so it helps me to maintain control of the vehicle. It's not getting beat to death in the surf. Makes Mike nervous when I do that, but gives me a little more authority to keep Atalanta pulled off the ship. Okay, Jaden, you can turn Atalanta's lights on, please.
Copy.
Control deck, that's Atalanta in the water.
Hercules is dive, dive, dive.
Roger that, Mike. Thank you. Hercules is dive, dive, dive.
Roger.
Copy.
Tether ups are zeroed. I couldn't get it going. Maybe I'll have better luck. Tracking action? That would be good. Yeah. Look at that. She can't hold her heading. I got good visual on tail to tail, so we're still not yet. I'm not quite deep enough yet. Only 23 meters, but get a beautiful view of it in Atalanta's aft camera. And compasses are opposing, and Atalanta's still somewhat on the vessel heading, so we're all good. Still in the box. They are on. They should be on.
The WCAs?
Oh, the cameras. Yeah, stand by, Amber. Wait till we get to 50 meters. Yeah, I'll pause at 50 meters, and we can take a breath here. And the butterflies are. They're not flapping and trying to beat their way out of my tummy at the moment. They're still pretty flappy.
control deck. I'll stop by 0 meters. Control's been sent to the van.
Give it a bump, Jaden.
Copy.
Roger that, all good.
Copy, control. We have control here.
Okay, let me just want to do some checks here, make sure I maintain that delta. You can tilt your Atlanta camera down a little bit into the light. I'm just going to light up the arm here and
Copy.
make sure it's all good.
It's an audio slate for dive H2080. UTC time is 22:25:45 mark.
Green lights everywhere. Ooh, that motor is hot, hot, hot. Okay, Jaden, you can start heading down whatever meters a minute I'm making.
Copy.
I'm good, can hold. Oh, why didn't you come on? That's weird. You got your lights on, Jaden?
Yep. Not all of them, but
Oh, that's better. Thank you. Yeah, it's got a bit of a mind of its own there. Look at us in the breeze, heading off to the east. Where are you, winch alt? I like to have the winch alt up there too, just so I can see it out of the corner of my little spy with my little eye. I'll just strafe the gauges real quick. Gauges are good. Roger. You all happy with the WCAs, are you? Roger. Gauges are good. I'm going to put this back on the craft, so if it does the salute, yell at me and I'll turn it off. Off comms for just a minute here.
Good morning from Palau. It is 7:30 Palau time. We are descending our vehicles into an unknown seamount that was mapped for the first time yesterday. The seamount is located approximately 86 miles southeast of Mirror Island and Summit, and we're going to summit at the depth of 545 meters. Yeah. So this dive track was chosen to traverse a variety of different habitats and depth range. Dive observations and samples will center on understanding and presence and distribution of marine habitats and high conservational value. Let's start with introductions from the front row. So let's go with Chris.
Yeah, sorry, we're still having some
Oh, okay.
operational talks
All
up here.
right. Sorry about that.
Yeah, no worries. We'll be back in a minute.
Okay.
All right, we're all set up here in the front row if you want to continue with intros.
One quick request before we do that. Can you update the time to bottom calculator? It's still set from the other dive.
Roger. That is good.
1,050 meters bottom target.
1050. We're taking it a little slow here.
Yeah, understood.
Just so we get some weight on the wire. And down to the, I like the 500 meter number, but 200 is good in a pinch. 250. Once we get a little weight on the wire, the sheave won't be flagging as much there, and hopefully. Is that for me? Yeah, so we launched kind of, maybe it wasn't for me.
Yeah.
Ready? All right. So welcome back. This is our 4 to 8 watch. We'll be off shortly. We'll start with introductions. So we'll start from the front row. Chris.
Hey, I'm Chris Krasnowski. I'm here today as a navigator and mapper. My day job is kind of as a consultant for developing ROV hardware and software, and also doing sort of specialty mapping work. So.
All right. Thank you for that. Let's go down the line. Go ahead, Dan.
Hi, good morning, good afternoon. Dan, sitting in the Herc chair. Grateful to be here, grateful to be in the water. This is my day job. We had kind of a spicy launch there. We were on the edge of our weather envelope. Current is about 1 knot, and wind is about 20 knots. So if you can see the deck camera there, the angle of the overboarding sheave is a bit excessive, and we already have 100-meter layback, so we're kind of watching the numbers here, and hopefully we'll be on the bottom in about 40 minutes.
All right, thank you for that update. Let's go with Jaden.
Hey everyone, I'm Jaden. I'm the Atalanta co-pilot for the 4-8. My day job outside of this is I work as a mechanical engineer and surface vehicle technician at the University of South Florida.
Thanks, Jaden. All right, Amber.
Multitasking, one moment.
Okay. No worries.
I'm here, okay. Hello. Oh, look out. Here it is. Hi, I'm Amber. I'm video engineer on the 4-8, and when I'm not on Nautilus, then I am a cinematographer based in Los Angeles. And next to me is...
I don't know if I'm supposed to turn around.
Yeah, there you are.
Hi, I'm Taya Jagadatz-Pollock. I am your video engineer intern, and my day job is junior in high school, so.
Welcome.
Yeah, welcome to the 4-8.
Yeah.
The best watch, even though we still don't have a name yet.
Someone said we should be called the sundowners since we always miss the sunsets.
But we watch them digitally.
Oh yeah, we watch some digital sunsets. Yeah. All right. What about Brian?
Hello, everyone. I'm Brian Kennedy. I'm on shore. I'm the chief scientist at the Ocean Discovery League and a visiting researcher at Boston University. I study coral and sponge distribution on seamounts and kind of ocean exploration and ocean exploration technology broadly. And I am the watch lead.
Thank you. All right, Harold.
Good morning. My name is Harold Carlson. I'm a grad student at the University of Hawaii at Manoa. I also study deep sea ecology, and I'm in the science chair 4-8.
Thank you. All right, Quinlan.
Good morning, everyone. My name is Kinlan Yao. I am the data logger for the 48 Watch. Off ship, I am an undergraduate student majoring in STEM at Palau Community College.
Very nice. Thank you. And I'm Kim Etsity. I am sitting in the SciComm seat. My day job is an educator back home in Arizona. I teach chemistry and biology. Good to have everybody back.
Okay, Jaden, I think we can pick up the speed a little here. I'm going to make an adjustment. So Herc's horizontals are just barely whispering, and full vertical down. Should bring my speed up to about 30 meters a minute.
Copy.
And Brian, whenever we're ready, can you go ahead and introduce the dive plan for today?
Sure. So we're diving on an unnamed seamount southeast of Mire Island in Palau. And this feature is one of those great examples of why satellite altimetry shouldn't really be trusted. So, you know, in the seafloor we have a map of the entire global seafloor. You've seen this on the internet or whatever, but it's based on measurements of sea surface height from satellites averaged over a long period of time to infer what the bottom looks like. And it's only got a resolution of several kilometers on a good day, and on a bad day it misses whole seamounts. So the kind of what we have in terms of global distributions of seamounts and shape for bathymetry is a best guess. So, and this is a really good example of this. We had a feature that looked like a conical kind of, you know, pointy top seamount that, depending on which version of satellite data you looked like, ranged from about an 800 meter depth to a 200 meter depth, depending on exactly which, you know, estimate of seafloor bathymetry you use. So we mapped it yesterday and discovered that it was nothing close to a pointy seamount. Instead was a ridge geo hybrid thing that's 12 kilometers long. It's not quite flat top, but it's not quite pointy topped, and has a shallowest point, has multiple shallow points, shallower than 600 meters. And it's also really weird in that the long flattish kind of part, it has peaks south of it that are actually higher than the shallowest point. So if this was subaerial, meaning above the water line where it had been eroded down like you think in a geoformation, I would have expected the points to have gotten taken out too. So this is a very, and it has these really weird slump scar features all over. This is one of the most complex seamount structures I have ever dove on. And it was one of those fun moments of watching the multi-beam paint the seafloor yesterday as it revealed a feature nothing like what we expected to find based on the satellite altimetry data. So we had a very hard time picking the dive site because there's like 10 different spots we want to explore on this feature. It's so complex. So we've got this kind of strange dive track planned that we probably won't be able to finish because of the weather conditions today. Our dive is going to be a little bit shorter than originally expected, and at any point the conditions could exceed our operating envelope and we might have to end the dive. So we're going to be a little bit on the quick side and try and cover as much ground. That being said, I expect some 100-meter wall faces in here, which we'll have to be even more cautious with given the weather. So while we want to make tracks, it's going to be a hard dive to make tracks on. So stay tuned and bear with us if we have to pull off the bottom for a little while or if we have to end the dive early.
Thank you, Brian. With such a short dive, do you think, or what would be our priorities today?
Well, we're going to prioritize getting the eDNA samples for our partners at Pickrick. We're going to prioritize getting a rock, and we're probably going to be a little more aggressive on sampling things that we see, but when we see them. Sometimes we wait for a good spot to get one or kind of debate and move on and see how many of them we see. And today, when we might have to leave the bottom at any time, we're going to probably be a little more apt to pick up anything that looks new or unusual and then make tracks.
All right, thank you for that. Also on our 4 to 8 watch, we have Jeff. Jeff, do you want to introduce yourself?
You can tell the excitement is high when the off-duty scientists start hanging out in the control room.
Orbit itself?
Oh, sorry, no. Duh. I was waiting for the Ethernet button to boot up. It would help, eh?
That's the button. Okay.
That button there. I'll also turn on the
Can you hear me now, Kim?
Yep.
Sextant digital stills camera.
All right. I'm Jeff Drazen. I'm not on this watch, but I'm here just to see things anyway. And I'm a deep sea ecologist and specialize in study food webs and fishes.
Thank you. What watch are you on?
I'm on the next watch, so
okay,
I come
so
on in
you'll
a few
be on.
minutes.
Okay.
Yeah. But I like seeing the stuff in the water column, actually.
Yeah.
In that case, we will adjust Atalanta's camera to get a little more water column action.
Thanks, Dan.
I can do the same for Herc's camera if you wish. I turn on those downlights that'll highlight those.
Okay. You ready for move back towards the point?
Yeah.
Bridge nav. Can we move 200 meters due north? We can do 0.3, that's fine. 0.5 is probably fine too. Okay. I don't think we have to worry about being able to go that way, speed-wise.
Yeah, we have to worry about going too far that way. They we got less of a flag on the aft wire, but still a significant one. That's the current pushing us off to the east there.
The USBL isn't being super great. It's probably lots of noise from the DP today.
Yeah, it's working. It's working today.
Yeah.
20 knots. That's over 20 knots now. Got some 2-meter heaves on the A-frame there.
Neat.
Huh?
Neat.
Yeah. Living on the edge. That's where we like it. Spicy. Really looking. When are we going to hit the 100-meter cliffs on our watch? Or?
You're going to be lucky if you get to the bottom on our watch.
No, I mean, well, our next watch, assuming we get one.
Yeah, probably.
Awesome.
But they're between, they're basically right where you can kind of, on high pack, that bottom left corner is about where they're going to start showing up.
Yeah.
Yeah.
No. Oh, yeah. Yeah.
Somewhere, yeah, waypoint 2 is supposed to be the top of the cliff, roughly.
Yeah.
Cliffs.
Look at this guy. Why didn't we go to that?
Cliffs and canyons.
We discussed it.
Dan would have loved that.
Oh, yeah.
I mean,
You would have made his day.
we would honestly like to spend the rest of the cruise on this feature, to be totally honest. This
All
feature
right.
is crazy.
Sick feature, bruh.
It is.
Assuming you've been in Hawaii too long. Assuming it's not mud.
Assuming it's not, well, I can
It'd be
also
some pretty steep mud.
I can promise you the stuff just north of waypoint 10 is not mud.
Yeah, it looks pretty rocky. What drove the decision to go where we're going as opposed to the
widest range of habitats possible in a dive? So we're gonna get deep, get up, see some steep territory between 1 and 2, then get some flat mud between 2 and 4, and then climb up another steep peak a little tiny peak that should be up in the water and should be coral heaven between 4 and 5, and then bonus points if we start getting past 5.
widest range of habitats possible.
Dan just wants to see sheerest cliffs possible.
Truth.
Cliffs and canyons, but it can't all be a joyride. There has to be some science in there too, which does involve mud.
Occasionally. Well,
occasionally.
Often.
Well, for those people like fish, we're blaming it on the fish people.
Yeah.
Looking at you, Harold.
I like herding fish too. That's quite entertaining. Although I've yet to catch one, to my complete embarrassment.
Gotta change that, Dan.
I mean, I didn't want to say anything, but
Yeah, Dan, this has not been a good cruise for you in terms of the being eluded by mobile creatures.
Yeah, it has not. And it's been good for Josh. He's got the only two, I think, slurp fish on the fly. I think we managed to slurp one early on, but it was like just sitting there waiting.
I believe
easy mode fish?
Yeah, easy mode fish.
I believe we have increased Nautilus's entire fish collection count between these two legs by 150%. So.
Yeah. We have quite a few fish.
Yeah, the previous count was only 6, so. And what are we at now? Definitely more than 6.
Yeah, we got
6.
6 on last leg, and so 6 plus 2 or 3 this leg, I guess.
Doing pretty good.
I usually get in trouble for harassing fish or herding fish.
Now it's encouraged.
Well,
It is
fish are valuable
encouraged.
samples too. Now we know they can be collected.
Has the net worked on any of them?
Depends
No.
on your perspective.
Right. Has it been part of the collection process?
I think Josh got something.
Sea cucumber
Sea
that
cucumber.
escaped was in the net.
Oh, that's
Yep.
heartbreaking.
Dan, when we get the chance, could we scooch the Orion arm a bit?
Roger.
Thank you.
I have a button for that. Button. Let me know when it's, not sure if it's even moving.
Keep going a bit.
I should probably look at it here. Give me a second. Yeah.
Looks like our next watch team's coming on in.
That should be out of the picture there.
I still see the clock.
Roger. How about now?
Just another inch or two. Just
Oh.
a bit more.
Really?
Upper,
still see
upper
it,
arm
the
is
upper
still
arm.
in there.
Oh,
You're
it
gonna,
must be the shoulder. Let me
yeah,
see.
you're going to have to rotate shoulder. There it goes.
I just had to pick up the shoulder a bit.
There it goes.
I still have the joint of
Oh my God.
shoulder, so sorry.
I
I have it on PC4 if you want to look.
No, it's more fun than listen to Harold.
It's still there.
You still have which joint?
The upper shoulder joint, I think.
Really?
Or the second joint? I don't know.
So it goes azimuth, shoulder, elbow,
Okay.
wrist roll, wrist pitch,
Oh, yep.
wrist
Then I
rotate.
think it's whichever this one is.
That would be pitch or wrist pitch.
Oh, yeah, I guess it's just barely there.
I know.
Okay. I still see it.
That's really weird because there's azimuth left and right.
May log up.
There's shoulder
Are you sure?
down.
Yeah, I see
And
what
up.
he's seeing.
No, I see what he's seeing too, yeah.
There's
It's-
elbow
It's still
No, it's
just
moving,
peeking
you're
out
right.
in there.
No, it's
Up.
moving.
and down.
I mean, we can always crop it.
There's pitch out, but
Okay, that's like,
oh, I know what you're seeing. Let me try this. That should be more better.
I mean,
No.
there's a couple pixels,
Roger.
but that should be okay.
All right, the 4 to 8
you.
watch team, we're out. We'll see you guys later. Have a nice day.
Um. Oh, hey. Bridge, nav, hold position. Oh yeah, there's the seafloor now. We're about 200 meters up.
Roger. Yeah, you can go static. We'll hand it over and let them creep down on it.
Want me to slow the winch down a little?
No. But,
Yeah, there she is. All right.
we'll probably come all stop on the winch when we hand over. So don't
Yeah, that's
don't get out
probably-
of the chair with that stick there.
No, we moved a lot, so we're moving back. But that's up to you guys in your watch if you want to get all the way back to that or not. I'm guessing they probably don't. They just kind of randomly select points, so.
Yeah, they do. They throw a dart at a nav chart wherever it lands. That's
Yeah, I'm already at 14 below you, so I will just give it a little bit. Now you can stick it at whatever you feel comfortable with, and I'll get it once we get kind of evened out. I'll be able to adjust.
Morning, everyone. I'm really excited for this dive today. Whenever you're ready, just let me know when you're good for introductions. All right. Okay, looks like we can start in the back. Ali, and good morning. My name is Amanda Dedicatoria, and I am a freelance writer and filmmaker from the island of Guam, from Guam. And I'm sitting here joining you as your science communication fellow.
Sure. Okay. Sure.
Morning, everyone. My name is Menzel Klematnir Meril, and I am from Palau. I work at the Office of the President. On board, I am the cultural liaison for you guys this morning. Thank you.
Ali, everyone. My name is Jeff Drazen. I'm a SWATCH team's lead. I'm a deep sea ecologist, and I focus on studying food webs and deep sea fishes.
Hello, my name is Greta Sartori. I'm a researcher for the Palau International Coral Reef Center, and I'm here in the Nautilus as a guest scientist and taking care of the eDNA project.
Hello everyone, my name is Noelle. I'm the data logger on this watch, and I am the science coordinator for the Ocean Exploration Trust.
And how about you, Max?
Hello everyone, my name is Max Luthi. I'm a video engineering intern on the Nautilus, and I'm in the video seat.
Go for it, Ray.
Sorry, my name is Rye. My name is Rye, and I'm in the Atalanta scene today. And sometimes I get those two things mixed up, so.
You are one.
Yeah.
Hi, everyone. Josh Chernov. I'm the Herc pilot for this watch and the ROV operations manager for OET.
Hello, folks. I'm Dan Torriello. I'll be the navigator for this watch.
Doppler.
Yep. 60 meters.
Awesome. Thank you, everyone. Jeff, would you mind just getting into a little bit about what we could expect today? Where are we? And why was this seamount picked?
Okay, I'm going to come all stop at 30.
We're about 75 miles. We're down in the Southwest Islands. We're about 75 miles southeast of Merrier. And we mapped this seamount yesterday and last night, and it is probably the shallowest feature that we're going to dive on. It's probably one of the shallower seamounts in Palau's National Marine Sanctuary, actually. The summit comes up to about 550 meters or so, and it's kind of a long feature with lots of steep cliffs and ridges on it, and so it looks really exciting. It does not seem possible to really cover this entire seamount, which is really large, in one dive. But we're gonna take it our best shot, trying to see what is here.
Okay, I'm coming all stop at 30.
Okay. We're going to end up descending to roughly 1,000 meters and ascend up the east side of the seamount till we get to the top. And there's a couple of little pinnacle features on top, and we'll try to make it up one of those.
Yeah, that's exciting. Thank you. So I have a bit of an announcement, which is pretty serious and, dare I say, mission critical. I lost my toothbrush the other day, which is very surprising considering this is not a huge ship, and there can only be like 3 places in which it can be, which are the 2 bathrooms near my room and like my dresser. So if you happen to see a blue toothbrush, I miss her. And my Sensodyne toothpaste, my delicate enamel, would thank you all. But so I've just been using like a little brush that you use to brush your reusable straws that I have left in my duffel bag accidentally.
Well, you're resourceful.
Oh, thank you. You know, does it work? Yes. Does it work well? No. But we keep going. So that kind of I wanted to like kind of ask the question, and I think it would be wonderful for you to get into it, Jeff, our explorer of the world and our seas below. You've gone on so many expeditions. Has there been an instant in which you were like, man, I forgot this one crucial thing, and I'm stuck here?
Oh, let me think here. There are some times when I've forgotten things. Mostly I don't remember the personal items, but I certainly remember the science things. I remember going out in graduate school on one of my first expeditions, and instead of leaving out of San Diego, which is what we normally did, we left out of San Francisco, but I forgot I was doing baited traps to catch rat tails for my dissertation work, and I forgot to buy bait. Oh. So I had nothing to bait my traps with, which means I would not get any samples. Fortunately, the cooks in the galley were very generous, and they provided a bunch of chicken, and trapping continued. So
that's amazing.
I am sure we've missed other things, but normally when we go out to sea, at least for the science gear, even my personal gear, we have checklists, and we go through them. So there is no store down the road. There is no easy way back to port. And here we are on seafloor.
That's amazing. Thank you for sharing. I'm sure, did they take to the chicken well?
It didn't work as well, no. Oh, no. And there's a little fish, a halosaur probably. It'll take a few minutes for these guys to get set up.
I forgot my fins while scuba diving one time, and if you guys are PADI certified, that's a huge no-no. But I didn't want to turn the boat around. Had too much pride, so I dove without fins.
Oh no! Oh my God!
And you swam hard.
Yeah, really hard.
Really worked those muscles. To our friends in the chat, I deeply thank you for all of your condolences. I will hopefully pull through this next week. Here's hoping.
How are things looking, Josh?
We're okay. We're going to do a bit white balance, and then we're just figuring out some nav stuff, so.
Okay.
Let's see. That seems a little better. Okay, I'm ready for white balance, Max.
Awesome. Can you turn off the lasers?
Sure.
I got them.
Okay.
Okay, I'm going to go for zoom.
Okay.
Okay.
Jeff, are you going to want to backtrack back to waypoint 1 here, or just proceed from our current position?
We're going to proceed from our present position. I think probably the first thing we'll do is collect a rock sample.
Great.
Yep.
All right. Are we good for the screen to go black for a second?
Yeah, I'm good.
Okay, going for black balance.
Sorry, I'm coming up.
Man, that always feels so long.
It's okay. I have 10 other screens to look at.
Okay, and we do auto white balance. Okay. Okay, we are good to go.
Okay. Let's come on wide. Are you feeling it right? You good?
Yep.
So Jeff, I heard first you want to collect a rock sample around here maybe?
Yep.
Okay.
I'm looking at this rock outcrop here on the right.
Is that crinoid moving?
Where do you see it?
Center screen.
Oh, it is. I think.
You're talking about the one that was on the coral? Just out of screen to above us?
Yeah.
Okay.
You just want to look at this little pile here?
Sure. Got a nice Metallogorgia. A couple of crinoids on it. Maybe you hadn't seen that it was attached.
I did not.
Yep.
So, Max, we have a question for you from the chat. When it comes to that turning off the cameras or the ROV lights, what does that have to do with the white balancing?
It's the auto white balance, auto black balance control, and that's just to make sure that our whites look white and our blacks look black.
All right, thank you.
A little big.
Oh, yeah.
Can you just pan?
Yeah, pan over.
Yeah.
Oh, there we go.
Ooh, there's a cool fish in Atalanta. I think it's a halosaur.
Okay. If we can shake a bit of the mud off and then do a spin in front of the camera, that'd be great.
You want to, oh, let me get the right, stand by 1 sec.
Sure.
Oops, I think I pushed something on your screen.
There was a fish in the background.
Okay, I can get it in the still cam.
Okay. Let us know when you've got a good shot, Greta.
Right.
Sorry, I think I touched your keyboard for a sec there, Dan.
Oh.
That's my bad. Sorry about that.
All good.
I think we have a shot.
Okay.
Maybe not in perfect focus, but... Okay.
Okay, that's good. If we can spin it in front of the video.
Do you want a laser size of that?
That would be great.
Okay.
Thanks for the reminder.
Let me find that second laser. There it is.
There we go. Excellent.
Okay, a little rotation.
Oh yeah, nice fish in the background there. Thank you, Josh.
Okay, we'll go starboard box, I guess.
Sure.
Let me know when you're ready for it to come.
Good.
Maybe we should get the DNA sample now as well, so then we're good to proceed with no more
You're
stuff.
okay, 1022. I mean, it'll be another 15 minutes before we're higher, probably, or 10.
Yeah, no worries.
We can make it exactly 1,000 if you want.
All right, let's do it.
Okay.
Let's do
This
the
going-
1,000.
Stratified sample.
I'm gonna try a little one on the back if it works.
All right.
Sorry, Jeff, were you asking to start moving the ship?
Let's pop up and look around for just a minute before we do that, Dan.
Okay. I was focusing on something else.
I've seen about 4 fish, and
yep.
knowing me, I'm going to want to chase one.
Sounds good.
There's also a little bit of science chat going on, and apparently this Metalogorgia rather is a different species than we normally see. So I've just
that
asked.
Was in D, Noel.
Got it. Thanks, Josh.
Just asked if we need to collect that.
That was great, Josh. And we just actually received a little shout out for you. It says, "Hey, Josh and team, it's Tara from LBCC."
Hey, Tara. Okay, I'm not going to put the arm quite away yet because it'll just drag it in the mud. So we want to pop up and...
Yeah, let's take a look around real quick.
Okay, I'm going to pop up, stow the arm, and get our
Sounds good.
Stuff back in where we like it for video.
Awesome. Thank you. So we have a question for Jeff that just came in. So if rat tails don't like chicken, what do you bait a rat tail trap with?
Usually mackerel. Squid works well too, but not as well as mackerel or tuna or something like that. Those seem to attract more of them. They ate the chicken too. They just weren't as avid fans.
Little picky.
Yeah, I guess they're a little picky. I think mostly that the chicken doesn't produce as big of a odor plume as fast, and so just not as many of them were as attracted as quickly. So
interesting. Thank you.
Yeah.
We know very well though that chicken attracts the nautilus in Palau. That's what we baked, the nautilus trap for the nautilus to collect.
That's interesting.
Yeah, I think they probably like mackerel and squid as well. But chicken definitely works well. Tested.
Oh, there's a fish right in front of us, right there.
Must be an exciting delicacy for them.
Oh, it is really close. What? I do not know what we've got here.
Zoom in, please.
Oh, this is a little... This is maybe a small synaphobranchidial. Almost certainly it is.
I'm having to back up pretty fast here. Oh, stop.
It's just drifting with the current, isn't it?
I'll see if I can kind of turn and let it go in front if it does that, maybe.
Nav, that sample number was 049.
Okay, we can try pushing a little more. Yeah, go for it. Yeah, see if you can get on there.
Wow, it is really small. Yeah, that's a synaphobranchid.
Do you think
but
it's
I have no way of telling which one it is. At least I can't.
Ooh, it's moving quick.
Is it a juvenile or is that?
Yeah, this is a juvenile. This cannot be a very old fish.
Let's see right through it almost.
Yeah.
Oh, yeah.
Great, thanks.
Okay, come on, please.
I love how none of these fish swim the way I thought they would. They all swim like vertically.
Looked like it was dancing.
Yeah.
Yeah, so it seems like the bottom current here is very similar to the surface current, which is interesting. It's not always that way.
Is it as strong?
It's not, I mean, it's there, but it's not unreasonable, I would say, as we're hiding behind the current right now. Wait till we get up to the very top, and then we'll have
Josh, we got another fish on the right down here, right by the rock.
Okay. Yeah, see it.
Nice.
I don't want to say what I think it is.
Yeah, you know what it is though now, don't you?
What is it?
Okay, let's... it's hiding. Okay, let's zoom in.
It's a halosaur.
It's a rat tail.
That's a halosaur?
Yep.
No.
Really?
Yeah.
Oh.
I can see the fin sticking off the back of the head.
Okay.
And the dorsal fin right in the middle of the body.
It's tiny.
Yeah, it is tiny. Actually, if we can get a good zoom on it, I will want to collect this one.
Oh, really? Okay.
Yep.
Challenge.
Well, it's
all right, let's try and... Is that all the zoom you got, Max? Or?
No, I was trying to be conservative with it.
Yeah. Oh.
Oh, yeah, definitely.
Oh, okay,
Really
now
dark.
I can see the halosaur, yeah.
Oh.
Okay. Like the nose there.
Yeah. Really dark.
Really dark, yeah. Okay, well, if we're going to try and collect it, we should
Let's do it.
Try and do that.
Let's do it.
So come wide, and then, Jeff, if you could kind of keep an eye on it.
Will do.
And then if you want to get the arm out.
Yeah, can you get that?
And I'll get the camera kind of set up.
That was a great
It's
follow,
going up.
Josh.
He's headed upwards.
It's like right in the
049.
He's charging you.
Okay, I'm getting suction.
Okay, I'll just show you the suction here.
Yeah, that'd be great. Okay, this is going to go in sample jar 1.
Oh. Just 1 sec, I gotta try and get it back in view here. Why am I gotten...
I've lost it.
Hold on a second. I've lost thruster control. What's going on? Oh, never mind. It's me being stupid.
Okay.
Shoot. Okay, hold on a sec. Let me look up. Just looking in the wide.
Oh no, okay.
I kind of backed right up into the dust there. Maybe we'll do, so if you're going to want to try and collect a fish, maybe we'll just get like partially set up, and then that'll avoid all this dance.
Sure.
So let's grab the, sorry, we may not, whoa,
Yeah,
did
that
get
fish
jammed up to
that fish is gone.
Okay, ready if I grab the, do you want to, can you extend out a little bit so I can see
I just had to adjust the PRV because it was at 100%.
Yeah.
Okay.
Okay, Barista DVL.
Sure.
Okay.
I'm not getting blue light. There we go.
Can't be right.
So I think kind of get where you're like holding it, and then we'll just sort of hold it down in that area, and then we can look around.
Yeah.
We'll keep the blue light on and just keep it indexed.
Sounds good.
I don't think.
Pick up here.
Can we still see the fish? So yeah.
Sorry, I'm having issues.
Sorry if I do another reset. It's jumping all over the place here.
It's not moving. We're right.
Alright, let's see for a sec.
I feel like everything is really slow compared to like my movement on the controller is not proportionally the same as it has been before. Okay, sorry, getting in there. Yeah, wow, weird. Okay.
Okay, I think that's kind of good enough there. Like, just keep it out to the side so you got a hold of it.
You want it more over here?
Yeah, just so I can still kind of see.
Okay.
Looks like a little Norella there or something like that, that coral.
American, come down.
Couple fish
Oh, look
there.
at that.
Yeah.
Okay. Let's see here. let's just take a zoom right on the one closest to us.
All right, let's zoom in, please.
These look probably like more halosaurs. That is correct. Really, really dark one. If we can get another zoom, then maybe we can try collecting for this.
Yeah, let's go for it.
So what do you
This looks like the same thing we were just trying to catch a moment ago.
Okay, it's really backing up, so we're going to have to, before I get out of
let's do it.
Position. Okay, let's go,
Great,
please.
Thanks.
What do you find interesting about halosaurs?
They're
can you come full wide?
Definitely abundant in Palau. There's quite a lot of them. They are.
I gotta turn on the suction.
They're sort of browsers across the seafloor. They consume small crustaceans, even pick off worms, things like that. And they are actually reasonably closely related to eels, but they are not eels.
Okay, let's do like a partial zoom.
Oh, interesting.
Keep the porch in view. Great.
Suction on?
Yep, suction's on. Down a little bit. It's
oh, that was close. He's up and to your left just a touch.
Yeah.
He's going
up,
rising
up.
higher.
Oh, come on, stop that.
There he comes. There he is.
Nope.
It's a wily one.
Oh yeah, I can't go up and down as fast as you can. Just gonna switch to this one.
I think there's a fair few of these, so.
Oh, we got another shot here.
All right, okay.
Wow.
Oh, suction is on full, yeah?
It's on full bore, yeah.
Yeah.
Come
on. He's a stronger swimmer, a little bigger. Oh, now I've lost it.
So how long do these halosaurs live? We just got a question from one of our
It
viewers.
looks like there might be a second halosaur just to the left of the lasers on the left screen going down screen. I don't know if you can see it. It's pretty dark, but that might be bigger though.
All right, let's go back to the seafloor. Okay. Yeah, we don't actually have great age and growth data on halosaurs. I think there's one study from the Caribbean using whole otoliths, which can have issues, but it was the best that was done at the time. But generally, what we know is that deep living fishes generally grow slower the deeper you progress into the ocean. So most of these fish are growing quite a bit slower than species you would see up on a coral reef or in shallow water. And it's not uncommon for species that live at 1,000 meters to live for many decades and not reach reproductive maturity until they're about 15 years old or so. So probably relatively slow growing.
Thank you.
Okay, I can take over the winch.
Okay.
One of the best examples of this is like the Greenland shark, right? That can live up to 400 years.
Wow.
So yeah, pretty much when Michelangelo was painting the Sistine Chapel, that shark was already alive.
Wow. That's awesome.
It's crazy.
Manzel, considering that we're kind of exploring this new area of Palau, are there any sort of stories or kind of legends that about the significance of this place?
Yeah, NavG's not working right here. Let's wait for that. Sorry if we pause for a minute. I'll go back to the USBL and see if we can start the DVL again.
Okay.
Okay.
Jeff, there's a fish in Atalanta Cam. Is that one that you...
I
I'm
saw it
not
too.
seeing it, sorry.
It looked like a little bioluminescent hatchetfish.
Oh, wow. Deep for a hatchetfish. We're like 1,020 meters.
Maybe not a hatchetfish then. That sort of shape though.
Round.
Yeah.
And reflective?
Yep.
That sounds like a hatchetfish.
So we're at Merir. Merir is a part of Sonsoro State. This is one of the southwest islands of Palau.
Can we zoom in, please?
And this would actually be considered within the
Cyclothony.
the contiguous zone of Merir. So
that's great, thanks.
Go, come what? Not interested in that one.
Sorry, you keep going, Manzo.
Specific to our to the way our culture is, the Southwest Islands have a slightly different culture than the mainland Palau. Mainland Palau is a matrilineal society, so we go by our mother's lineage. The Southwest Islands are a patriarchal society, so they go with their father's lineage. And a lot of the cultures here are actually similar to the Yapis from the Federated States of Micronesia. Similar, not the same, but similar. But very different from our own culture in Palau. Although we are considered one country, they practice a different way of life. Yeah.
Wow, that's amazing. Thank you for that.
Yeah.
Wow. Just using the ROV like a jungle
Very
gym.
active. I cannot keep up to that.
It was swimming behind the sample jar, and so it just looked for a moment like it was inside.
It's just not fair. Okay, as we come up, can we take a zoom on this coral right here?
It was just teasing Jeff.
Yep. So these really small fish, you can see another one down here. These are in a genus called Cyclothony. They're called bristlemouths. And this genus of fishes are the most abundant vertebrates on the planet. They live across very broad depth ranges from a couple hundred meters down to a couple thousand meters. And they live in all the world's oceans. And we could catch, when we were trawling, as many as 1,000 of them in a 1 hour tow. They can be incredibly abundant. So we're going to be ignoring those today. There'll be a little bit of noise for us. Bamboo coral here. Do we have any more zoom for the polyps?
Absolutely.
Yeah, go for it.
Okay.
All right, NavG seems to be working right now.
Okay, thanks.
That's great. Thanks. Well, let's keep heading up slope.
Do you want to move the ship up slope, Jeff?
Yeah, I think we should probably start heading up there.
Sure.
I'm gonna trigger it.
We can zoom on that coral,
Okay,
I
let's zoom in,
suppose,
please.
if we can.
Hello, Renee.
Pretty.
Yeah. Black coral? No.
Go, come wide, please.
That's Calyptrophora. Okay.
Could you just kind of rotate the arm out a little bit.
Yeah. How's that? Is that in
Yeah,
here?
that's better.
Can get it even more.
Yeah, that's great.
Okay.
Really tall whip over there.
Stream plus plus.
Cute. Oh.
Can we zoom on this coral here?
Yeah, I'm just gonna not. I just don't want to knock over the
Yeah,
room.
yeah.
Okay, let's zoom in, please.
Is that a ctenophore?
Yeah, there's a little ctenophore drifting in the corner there, bottom right. Got a primnoid here.
Oh.
Oh.
Hello.
That's that's one of the bathygattids. You can see the really long fin rays all the way around, dorsal fin, pelvic fins. Cool. Okay, we can keep going.
Okay, come wide, please.
That was a great transition there, Josh.
That's awesome.
Another coral hair too.
Yeah.
Is that one on the right? Is that a Chrysogorgia?
It looks like it. Yep.
Yeah, okay, cool.
But let's
let's keep
zoom
going.
in on this big
Oh,
one
this
right
one?
here for
Okay.
just a second.
Yep, we'll zoom in, please.
What's that?
Yeah, I'm looking at the thing on the left. Is that an egg case?
Is that a snail?
I'll come around a little bit. I'm going to tilt up a little bit. Let's see if I can pirouette around here.
I've got more zoom.
Okay, I'll get it stable. I'll come around. Okay, we'll zoom in. Yeah. I pulled on a little bit there.
I think. Yeah, this is probably an anemone.
Kind of bumping the craft arm there a little bit.
A lot of brittle stars, little
Okay, well,
crinoid.
come wide, please.
Oh, yeah. So we've got this is the coral is Thororella. We collected some of that a couple dives back, and the blob was a closed anemone.
Do you want to zoom on the... No, it's probably the
Any
same as
halosaur
before.
there again?
The fish.
Oh, I missed the
Down
fish.
there.
There was a fish back behind us, just below.
I think it was the same...
Oh,
one
is
we
it the
just-
bathygattis? Yeah,
No,
let's just we'll keep going.
I had that feeling.
Okay.
It's so hard to tell the difference between them when they're small.
Could we look at the other set of gauges, the res and the main?
Yeah.
Great, thanks.
Let's go take a look at that over there.
Bridge nav, we can go another 20 meters at 264. Thanks.
Ooh, that's a big fish.
Oh, yeah.
Oh, yeah. That's a cutthroat eel in the back. Let's just focus on the coral, though.
Roger.
Can't believe I just said that.
Flash from me.
We have enough fish at this point.
Hey, let's zoom in, please. Those are crinoids.
Okay, and they were all together.
All together, which
yeah,
look like,
it just looked like a really funky, you know, Canthogorgia or something like that at a distance.
Slide over here.
Okay. Yeah, can we zoom in on this primnoid here?
Yeah, we'll zoom in, please.
What is a primnoid?
She asked the fish guy. It is a large family of corals, and I am not entirely certain how to distinguish them taxonomically or ecologically from some of the other corals, such as bamboos or things like that.
Come on, please.
Yep, that's great, thanks.
Okay.
It's pretty.
It is. Paragorgia, maybe? The red one?
Yeah, probably. Sorry,
Okay.
Ryan, could you do me one more favor?
Yeah.
Could you just pitch up and
Yep.
hold the arm like that? Because I just, every time I get down low, I keep bumping the
Yeah, no problem.
into the net. It keeps pushing me away from the zooms. Yeah, awesome.
You good with this?
Yeah, that's great.
Okay.
Thank you.
Yeah, no worries.
All right, can we zoom in, please?
There sure are a lot of these here. Okay.
Okay, come white.
Let's head up slope.
I'm just going to take a look at the Keist con.
Okay.
Hello, Ocean Explorers.
Who is that? A voice from above?
It was God.
Yeah, God, is that you?
What have I done? And what can I do to rectify it?
It could be Ecolau.
I lost my toothbrush, please. I've suffered enough.
Was that Ecolau?
Oh.
I think it was Rachel from the data lab.
Oh, hi.
Okay.
Hi, Rachel.
Yeah.
Hi, Rachel.
We were looking to turn on grammetry on both cameras. I think we've fixed most of the issues with it causing problems on causing the video feed to stutter, but we just wanted to check with the folks up there about turning it on the upper camera.
Okay.
That's your call, Josh.
Sorry, Rachel, what did you want to turn on?
Rachel, can you hear Josh?
She wanted photogrammetry.
I'm on the data lab, by the way.
Yeah, sorry, Rachel, I didn't hear what you wanted to turn on.
Fish. Huh.
I'm talking to data.
Yeah.
Do you?
Rachel, can you hear us? Anyone?
Rachel, can you hear Josh?
I don't know if she can hear anybody.
I'm talking to data, but
Okay. Yeah.
and I'm talking to SPL.
She wanted photogrammetry turned on the upper camera.
Oh,
She thinks she resolved most of the problems with the feed stuttering.
okay, she can do that, but I don't think I have control over that. Yeah, sure. If she can hear me. Yeah, Rachel, if you can hear me, yeah, go ahead.
Okay, cool. So yeah, sorry, I didn't have this intercom panel set up right.
Okay. Yeah, so you're going to do test some photogrammetry with the upper.
All right, yeah, and if there's we have panels linked to SPL down here, so if there's any problems, just call out and we'll turn it off.
Sounds good. Okay, we can zoom in, please.
So there's two different, this is a cutthroat eel, and there are two subfamilies, and pretty much two common genera in these kinds of depths, Synaphobranchus and Iliophus. And we can get a side view of the head, we can tell which one we got. But they're very fast.
Okay, I'll try it again. Let's go for zoom. Oh, yeah. It's
Whoa,
yeah,
okay.
Taking off.
Oh, no, that's not bad, though. I mean,
Sorry.
just for ID purposes, not cinematographic, but...
yeah, not cinematographic.
Okay, let's keep going. We'll end up chasing this thing all day.
So Dan, as a man of sound, science, and sick beats, I am pleased to announce that you have received a fan message from the chat. It said,
tripod fish right there.
Tripod fish.
Oh, yeah.
Sorry, I'm just like way behind her.
It's all good. Intermission. Fish intermission.
Quick zoom and then we'll keep moving here.
Ooh,
oh,
it's pretty.
Great. Yep. Nice tripod fish.
This one looks different than the other ones you've seen.
It is. It's got shorter fins. Oh, wow. Look at the really tall rays sticking up off this bottom from the pectoral fins. They branch part way up. You see the little wisp-like fins? That's how it's detecting its prey moving down current towards it.
What does it eat?
Almost exclusively small plankton, copepods, things like that.
I'm going to have to keep going here, guys.
Yep, no, that's great, thank you.
I'm close to the seafloor to give you more tether.
Okay,
so.
I'm going to go up.
Yeah.
Tripod fish. Let's see. 4. All right. So, Dan, you got a fan message from the chat. It said, big shout out to Daniel. I watched several of his band videos last evening. Smooth and enjoyable. Would enjoy his band in our community music venues. We have a valley of live music, which I prefer. VT.
That's a very kind comment.
Yeah.
Thank you.
And to our friends in the chat, if you didn't know, Dan Toriel, our very own navigator, is an amazing drummer, and his band is called Harsh Armadillo. I listened to some of the songs, and if you could hear Sunset, that would be it. Highly recommend.
Excellent testimonial. Thank you.
We only do peer-reviewed stuff here.
Whoa, big fish top left.
Whoa.
Yep.
Zoom in, please.
Wow. What is that?
That is another cutthroat eel or Synaphobranchidae. And looks like Snaphobranchus. Probably Snaphobranchus brevidorsalis. The dorsal fin starts fairly far back on the body. Okay, let's keep going. Thanks.
Please.
So Rachel is God around these places?
Yes, because she helps us with our Wi-Fi.
Okay, for the DVL reset.
It's one of the super common
And so many
Oh yeah,
other things.
never mind. Skip that one. Sorry.
Just about back out front, just getting there.
So we have a question from one of our viewers in the chat. Why are so many fish in these kind of deep sea areas have like a sort of eel-like shape?
The pressure. Just kidding.
We've got a lizard or a tripod fish on the left, and this is one of the bathygattids right here. Oh, this is great. Look at this. You can see all the fins. That's basically its radar dish, so that it can sense things swimming up ahead of it.
Its face is
Can we
so
turn the
interesting.
lasers off for a minute, actually?
Yeah.
Sure.
It looks like a satellite from this angle.
Yeah.
Doesn't it?
Sputnik.
So we did collect a couple of these, so I can't quite identify it to species with the information I have out here, but we'll get some species IDs on these fish later.
Staring right at us.
Very reflective.
Yeah. Its eyes are full of fear.
That's great. Thanks,
Okay,
Josh.
Light, please.
And can we get the lasers back on?
Yeah. Thanks for reminding me.
Yeah, I always forget.
Tripod fish.
Yeah, we'll keep going. It's the same one as the last one. We have a bit of an abbreviated timeline for this dive, so I'm trying to be good and keep moving.
What are these mounds that we've been seeing?
A lot of these mounds are the burrows of worms. They're deposit feeders. They're eating detritus in the mud, and so they're taking up mud probably from the surface. And then their tail is located some distance, a little bit of a distance away, a few tens of centimeters, and that's where all the mud ends up in a big pile at the end. Yep.
What was it, 264?
So
there was a question about fishes and these eel-like tails, and I didn't get a chance to answer that because we were seeing a fish. A lot of the fishes we need to take a eDNA sample here. Almost forgot. We're right at 1,000 meters.
Okay. You want to do that?
But these fishes end up with these long tails, mostly because that form of swimming, the undulations along a long skinny tail, turns out to be a very slow way of moving your body.
Red ctenophore right there.
But it's, I see it there. But it's very energy efficient. And in the deep ocean, where it's very dark, these fishes typically don't have as much need to be able to just swim really fast. So they don't end up with paddle-like tails. They sacrifice that for that consistent high speed for just efficient slow speed swimming. So you see it in the rat tails, you see it in cusk eels, which are not eels. You see it there's a lot of eels down here as well, actual true eels. The halosaurs have that long eel-like tail.
Yeah.
Thank you.
I'm going to perch back in.
This isn't a strategy you could get away with in shallow water. If you had that long eel-like tail and couldn't swim fast for a good period of time, something like a tuna would just eat you. So, yeah.
Yeah, thank you for that.
Sure.
Oh, that's tiny little, okay, it's gone.
It's nice and slow.
I think it's worth doing. This
Yep,
is
popped.
050.
Niskin taken.
Thank you.
That was number 1.
Thanks.
Sure. Okay. Yeah. Yeah.
So,
Sure.
Greta,
Yeah, I understand.
when you process your eDNA samples, how do you keep your own DNA
Okay. Yeah,
from
you
getting
can just
to like
turn that
the
guy.
process?
So we take a lot of precautions for avoid contamination. We wear gloves. We use a substance called
gets, like,
RNase
stuck.
away, which actually dissolves DNA, and which we use it to spray our gloves
like a tool
periodically,
just to get it to move a
so
tiny
to
bit,
make
and then
sure
you can
that
do
we
it,
don't
yeah.
contaminate the samples. And also we make sure to, at the end of each day, bleach everything that we have used, so that we decrease the chance of contamination. It's not a tragedy if the samples get contaminated with human DNA, because we know humans don't live in the deep sea. So if we find some human DNA in our samples, we just don't consider it and we just look at the fish. Make sense?
Yeah.
All right.
Awesome. Thank you.
You're right.
Are we still? Yeah, we're still stepping. We can put another one in if you want.
So, Josh, we got a question from one of our viewers. What's happening when you pull on those like numbered balls on Hercules? Like, how does taking an eDNA sample work?
Well, we're just actually collecting the water. So we have 6 5-liter Niskin bottles, which are water collection bottles. Essentially, there's 2 kind of cap plug type things on each end of a tube, essentially, and then they're on a trigger. So we have them set up to you just pull on those balls, and that releases the trigger, and then both the ends of the caps close. And then you capture your sample of water at a unknown location.
Do we want to be set up for
fish
suction
right there
again?
on the right.
Or, okay. Are you okay if I do the blue button?
Yeah, let's do that. Yeah, thank you.
Yeah.
Thank you.
Yeah, let's zoom in, please.
Have we got any more zoom?
You got any more?
We do.
Max? Yep, let's go for it.
A lot of little orange amphipods crawling around in the branches. Oh, there's a mysid swimming off on the right. Great.
Okay,
Thank you.
Light, please.
And coral like that just never really looks quite in focus. They're so fuzzy.
That was Calagorgia.
We could just keep our momentum going when you're ready to step. Yeah.
Ali and welcome. We are currently exploring an unnamed seamount southeast of Meteor Island, which was mapped for the first time by EV Nautilus. This seamount summits at a depth of 545 meters, making it the shallowest known seamount within the Palau National Marine Sanctuary. If you have any questions, any comments, please send them over on nautiluslive.org.
There's something right there, Josh. See that little fish right there?
Yeah, zoom in, please.
Okay, this is another one of these little juvenile cutthroat eels. And it definitely has something in its stomach. It has just eaten. Okay, let's keep going.
Come
Thanks.
on, please. Yep.
Let's take a look at that right there. Looks like a sponge. Ferronematid, perhaps. Oh, there's a couple of them.
Fish in between them,
Yeah. Let's zoom on the fish first, because it's going to move.
It moves, yeah.
Yeah.
All right, let's zoom in, please.
Oh, this is a cusk eel. It looks like it's missing the tip of its tail. Alright, turn to the side, fish. There we go.
It listened.
It has something going on with its eye. Either there's a parasite near the eye, or Its eye is opaque. If we can get another good zoom of this fish, let's try to collect it if we think it will go up the suction sampler.
Okay. We'll zoom in, please. Got some more zoom?
I do not know what genus to put this fish in.
We might have to, yeah. We should
Really distinct lateral line.
Yeah, we should do that. Let's just try and keep it in frame, just like kind of, yeah, there we go.
Oh, great. Yeah. See a bit of a hook over the pectoral fin there on the operculum.
Oops, sorry.
Oh, no, that was great. Let's try collecting this fish.
Okay, we can give it a shot. Okay, can we just, sorry, Max, can we just come full wide again for a sec? Thank you.
Yep, no problem. Would you like me to just go full wide anytime we're doing suction sampling?
Yeah. It just kind of depends. Like, it's easier when I can see everything when we're trying to grab the nozzle and stuff, because then I can sort of get everything in frame there.
Copy.
Right. Yeah. Okay.
I don't, but I mean, if you have Wi-Fi, you should be able to access the stream video, so.
It's not coming through, Max. There's something's going on.
That would be a Rachel question, I think.
Okay.
Sorry.
Could you try to let Rachel know?
She might be listening.
She might be listening? Okay.
Yeah. I think she said she was at SBL on maybe too.
I'll radio data lab though.
I gotta go back to still cam. Okay, how you feeling?
Video, we don't have your...
I'm getting pulled on right now, I think.
Science portal.
Yeah.
Yeah, you can come down 4 or 5 meters probably. Yeah, we are, yeah. Sure. Okay, let's do a partial zoom, Max. That's great. Maybe back just a little bit more. Yeah, okay. You good with that? Okay. And suction on. Oh, now it moves. Go on.
All right.
I just wanted to creep up on it there and it just took off.
There it is.
What's that? Atlanta?
I don't know. Oh, that's it. Yeah, that's Atlanta.
Okay.
Oh, has a little more juice than I thought it might have.
Nothing to see here. Just
Everything's going so normal. Just another day in the sea.
almost touching bottom, so you might have to kind of reach out a little bit.
What's up?
Oh, it's raining.
There's a cup coral right below you right now. Or a
sponge.
Sponge, I mean. Oh. Oh, that's rain. I was wondering what that sound was.
Oh, yeah. It's wet here and below.
Cam bow is looking crazy right now.
Yeah.
Very rough.
Oh. Okay, maybe one more try and then we'll keep going.
Okay. It's pretty big. The bigger ones seem to have more fight against the current of the...
bigger they are, the more swimming power they've got, and yeah.
So weather is not cooperating, fish are not cooperating this morning.
Nope.
And I'm getting out of...
You'll just got to try your best.
Yeah, exactly.
Okay.
We're not gonna give up.
Yeah, work with what we have.
I think we're done right now.
Okay, we put the arm away?
Yeah.
Yeah.
Yeah, okay.
Yeah, let's put that away. Okay, we'll come wide, please. Yeah, the ship's...
Thank you for trying.
The squall
Appreciate
here
that.
is the ship's lost position as well. So we're going to have to get ourselves back into position here. Can I see the DP screen again?
Yeah, just an FYI, the Starlink's going to be totally inoperable until the rain stops.
Okay, thank you for that.
I am coming up a bit.
Okay, I'm trying to get on the other side of you.
Yeah.
Okay, thank you.
See if I can turn and go. Okay. Yeah, you hold your heading like that, and I'm going to try and pass you.
I've got auto heading on, so.
Okay.
If anyone's tuning in from mainland Palau, please control the rain.
It's Imaleek's people's fault, right?
Yeah. Anyone from Imaleek, we need the rain to stop.
Please. I didn't bring an umbrella.
Yeah, this was the risk when you go in and marginal. If it gets a little worse, then sometimes it can impact the dive a bit.
Yeah, it's basically, the rain's dense enough that both Starlinks as well as our traditional VSAT system are not able to get through.
Well, that's okay. There's not much to see at the moment.
Okay, I'm going to start bringing my heading back towards the ship, if that makes sense.
Yeah, I'll just I'll keep going.